Good afternoon, viewers. Today we are going to discuss a particular topic, that is applications of plant biotechnology. Now, this topic it is included under Economic Botany and Plant Biotechnology, BBYET 143. It is Unit 15, included within Block 4. The program is BSc General in Botany. So, in this particular session, we are basically going to talk on the transgenic plants, the genetically modified plants, and what are their implications or what are their applications so far as improvement in the quality of the crops or improvement in certain traits are concerned. So, we will be discussing chiefly on the way by which you engineered these crops, number one, and number two is what are their different applications in agriculture. Now, what is meant by biotechnology? We all perhaps know that in case of when you are constructing any genetically modified crop, then essentially what you are doing is you are trying to incorporate one gene. You are trying to incorporate one foreign gene into the plant system so that you want any desirable trait. You want any additional trait which we sometimes call as the value added trait. That means you want to introduce some quality or you want to introduce some novel trait into a plant system. Now, this kind of genetically modified crops has found immense applications in different areas like generation of abiotic stress tolerant plant, generation of biotic stress resistant plant, generation of nutritionally improved plants, generation of plants which would be very much tolerant or resistant to different environmental stresses, generation of plants which would produce some novel pharmaceutically active compounds. So, these are some of the different applications of genetically modified plants which we will take up one after the other in this session. So, basically this technology, it involves gene transfer or it involves introduction of novel genes. Now, the next point is that there are two ways to do this. Initially, when people did not have this technology, then people used to go for plant breeding. Now, in the picture that is given in this presentation, you can see that two techniques are placed parallelly. One is the plant breeding technique and another is the plant biotechnology technique. Now, initially what is meant by breeding? That means you cross, you hybridize. That is one plant having the desired gene or desired trait and another plant which is not having that trait, you cross and you bring the desirable gene into that particular recipient variety. But the problem with this technology is that here, instead of just adding one particular gene, you are getting gene clusters. You have the transfer of a cluster of genes rather than one specific gene. On the contrary, if you go for genetic engineering or if you go for biotechnology, then you can precisely select one gene which is your target gene and you can incorporate into the recipient plant. So, there is no question that the other genes will also be integrated into the recipient plant. So, that is why it is more advantageous to go for biotechnological approach or genetically engineered approach rather than breeding approach. Moreover, breeding approach also takes time, which we will be discussing shortly. So, what are the benefits of genetically modified crops? It allows a very wide selection of traits within a very short time. That means it is very fast and it is comparatively cheaper as compared to breeding. And you can obtain the changes in a very few generations, whereas breeding, it takes almost 8 to 10 generations or even more. Here it takes it is very fast. It may occur within maybe 2 or 3 generations, you can get the transgenic plant. So, it allows greater precision and it reduces the risk of random occurrence of undesirable traits. That means, as you have seen in the diagram before, that there is no question that the unwanted genes will also be transferred. Only a specific gene will be transferred through genetic engineering approach. So that's why people are nowadays, they are focusing more on these genetic modifications. Now the question is, how do you generate such a transgenic crops? What are the different steps? Now the first step is, first you have to know the gene. That means you have to identify the novel gene or the candidate gene which you are going to incorporate. So that has to be identified first through the different genomics tools. Second point is, you have to clone it in a desirable vector, and then you have to incorporate it into a plant system. In plants, we have the traditional protocol, that is agrobacterium tumefaciens mediated transformation, which I will not talk in details today. Maybe in some other discussions we can talk on that. So you use the agrobacterium tumefaciens mediated transformation to incorporate the gene along with the vector into the recipient plant species, and then you have to select those plants in presence of antibiotic or antibiotic resistance or antibiotic selection, and then you regenerate the transformed plant, and then you can screen those transgenic plants and finally see if your transgenic plant is showing a particular phenotype or a particular trait which you want. So those are actually the steps by which you can generate the transgenic plants. So what are the different applications? Because today our topic of discussion is mostly the applications of plant biotechnology. So as you can see in the presentation, that you have these applications, mostly, grossly you have these 5 or 6 main applications. One is abiotic stress tolerant plant, number 2 biotic stress tolerant plant, number 3 biofarming, number 4 biofortification, number 5 herbicide tolerance, and there are many others. You generate the male sterile plants also through transgenic approach. You can generate plants which are better capable of incorporating carbon dioxide, because nowadays CO2 concentration due to global warming, there is an increase in the carbon dioxide concentration. So those plants which can better use this carbon dioxide. So in that way there are several applications which we will take up one after the other. Now first, what are the different dimensions of abiotic stress? I think in one of my earlier discussions, if you have followed, we talked on plant stress responses, and mostly I dealt with the environmental stress response. So when I talk of environmental stress, basically there are osmotic stresses like the salinity, the drought stress. There are heavy metal toxicity, heavy metal stress, that is arsenic, cadmium, lead. There is extreme temperature, that is high temperature, chilling, etc. And there are many others as well. So here I have given a few examples. So what are the ways by which you can actually generate abiotic stress tolerant plant? Now, first of all, we have to understand that whenever there is a stress response, particularly when there is any form of environmental stress, then which are the pathways or which are the genes that get triggered? Now, if you remember, these points were discussed in the last lecture, that we have the induction of the osmoprotectants or the osmolytes. You have the late embryogenesis abundant genes or the proteins. You have the different antioxidative enzymes like superoxide dismutase, catalase, peroxidase, etc. And you have the different ion transporters like the sodium Na plus H plus antiporters, etc., to name a few, which are actually induced during stress. So probably the best response would be you go for the cloning and overexpression of any of these genes. Like for instance, I give you an example, proline. It is a very important osmolyte. So you have to fish out which is the enzyme that is responsible for proline production. So you can just clone that particular gene. Or if it is a reactive oxygen species scavenging enzyme like superoxide dismutase or catalase, you can go for the overexpression of that enzyme or that gene. So that is the approach. So you have to understand which are the pathways or which are the proteins or enzymes which are triggered, and then you can go for the overexpression. So in that way you can generate the different environmental stress tolerant plant. Now if there is a high temperature stress, then you can go for the overexpression of the heat shock proteins, which normally act as the chaperones, and their main task is to prevent the denaturation of the endogenous proteins. So you can go for the overexpression of any of the heat shock proteins. Similarly, if you want to induce chilling tolerance, then you can go for the overexpression of glycerol 3-phosphate acetyl transferase, which people have shown that they have cloned this particular gene from pumpkin, and they have cloned it in tobacco, and they found that the tobacco plants, they are more tolerant to cold stress. So in that way, there are different examples. So you have to select the gene and then clone the particular gene. In case of heavy metal tolerance, you have the phytochelatins, and the best thing would be to select the enzyme or the gene which overproduces phytogelatin, that is phytogelatin synthase. You can overexpress that particular gene, and you can in that way you can generate arsenic tolerant plant or cadmium tolerant plant, because phytogelatin is normally the component that actually sequesters or that actually quenches or scavenges the heavy metals. Now, perhaps all of us have heard about BT gene or BT transgenics. Now, what does BT represent? BT actually represent Bacillus thuringiensis, and we have probably heard about the BT cotton, which is one of the very well-known transgenic crops that has been developed in India, and because of this BT cotton, the cotton production has also increased a lot. Now the question is how this BT cotton is generated, or how the BT transgenic plants are generated. Now Bacillus thuringiensis, it is actually a gram-positive bacteria, and this Bacillus thuringiensis, it produces two types of toxin, as you can see. One is crystal toxin, which is encoded by the different cry genes. So cry means crystal. From the word crystal, cry comes. And another is the CYT toxins, that is the cytolytic toxins. Now we are more interested with the first one, that is the cry genes. Now you can see here that there are different cry genes, cry 1, cry 2, cry 3, cry 4, etc., which we won't discuss in much detail. The thing is that we are interested in the cry gene. Now you can see how this cry gene actually look. It has a pore forming domain and it has a receptor binding domain. So it is actually a toxin, but mind that it is not a toxin for humans. It is a toxin specifically for the insects. So if anybody says that BT transgenic crops, they are equally harmful to human, the answer is wrong. The point is, you have to remember that this BT is very, very specific only for the insects and not for humans or any other animals or organisms. Now, what is the mechanism of action of this BT gene? How does it act? Now, the BT toxin is actually lodged in a large structure which is called parasporal structure. Now, once it is solubilized, protoxin is released. Mind that this protoxin is not harmful or it is not detrimental. The protoxin actually needs to be activated, and there should be a cleavage so that it is activated and it exerts its detrimental effect. Now these crystals, these are large protein of about 130 to 140 kilodalton, and that is a protoxin. Now this protoxin remains as such, but whenever it is being ingested by the insect, that is by the insect larva, then it is solubilized in the reducing conditions of high pH, that is pH 9.5, and this condition is very much unique in the meat gut of the Lepidopteran larvae, that is in the insects. So only under this particular condition this protoxin will be activated. Otherwise, in other systems or in other organisms, you don't find activation of this protoxin. So what happens after it is solubilized in the insect gut? It is cleaved. So there is a proteolytic activity, and then only the active toxin, which is about 60 kilodalton and which we call as the delta endotoxin, is being released. and then it will bind to the midgut epithelial cells. It will bind to certain receptors which is present in the intestinal epithelial cells, like the cadherin receptor, the alkaline phosphatase receptor, the aminopeptidase N receptors, and then only it will actually exert its action. And ultimately what will happen? Ultimately it will create a septicemia, or it will create an osmotic lysis. So there will be a leakage of ions, and there would be the loss of electrolytes from the insect body system, and therefore the insect will die out of osmotic shock or osmotic lysis or septicemia. So that is actually the mechanism of action. And once more I repeat that this mechanism does not hold true for any other organisms. So BT is absolutely safe for other organisms. There is no harm or risk associated with BT consumption in other systems apart from insects. Now, do the plants have immune response? The answer is yes. Now, if you want to generate plants which are fungal resistant or viral resistant, or let's say bacterial resistance, because insect is not only the pathogen, or it's not only the agent that causes harm to the plants. The bacteria, the fungi, the viruses, they equally affect the plant system, and the plants are susceptible to these pathogens. So there should be some mechanism, or there should be some biotechnological approach by which you can generate this kind of transgenics. Now, in plant pathology, there is one important hypothesis which we call as the guard hypothesis. Now, what does this guard hypothesis means? Now, whenever there is a pathogen infection, okay, the plant first tries to overcome that pathogen infection, okay, just by the external defense mechanisms, like it has a cell wall, and it has different other defensive barriers, just as we have. Like in our case, it is the skin. So in the plants also, they have some outer barrier or external barrier by which they can overcome. But if the pathogen finally succeeds in overcoming that physical barrier, then the pathogen enters inside, and then the pathogen tries to release certain virulence factors, which we call as the AVR. And it secretes certain virulence factors, and in response to those virulence factor, what does a plant do? Because plant has to survive. So what does a plant do? Plant will secrete certain proteins, or it will secrete certain maybe enzymes as well, or proteins, and normally we call these proteins as R proteins. R stands for resistance proteins. So just like an antigen-antibody interaction which we have in our human system, what happens is this R protein, they interact with the AVR, that is the virulence factors which are released by the pathogens, and they try to nullify the effect of this AVR. So it is almost similar to the antigen-antibody interaction in our system. Then what does the pathogen do? The pathogen sees that, okay, fine, then I have to change my R gene. I have to create a new allele of my R gene. So it tries to generate a new allele or a new gene or a new effector, and the pathogen and the plant system, what does it do? It again synthesizes a new R protein. So this tug of war between the pathogen and the plant, it is going on in nature. And ultimately, who will win the race? That will be decided by who will synthesize more of these R proteins, or there is another cluster of protein which are known as the PRP proteins, or the pathogenesis related proteins. For example, chitinase. This is a pathogenesis related protein. Or beta-1,3 glucanase. This is a pathogen resistant proteins, PRP proteins. So you can overexpress again these kind of PRP proteins or R genes. Okay. So if you try to create a transgenic plant which overexpresses these R genes or R proteins like chitinase. So chitinase means it will degrade the fungal cell wall, because the fungal cell wall has a lot of chitin. So it will degrade the fungal cell wall. So this is another approach, and this approach is actually based on this guard hypothesis. So guard hypothesis actually shows that the plant actually tries to detoxify, or it tries to nullify the effect of the AVR genes or AVR proteins through the synthesis of different R proteins. So the best thing would be to go for the cloning of these R genes, as I have shown here, that identification of the resistance genes, and then you go for the cloning of the different R genes. Now there are different examples like BST gene from pepper, XA21 gene from rice. So these are the different examples. The list is endless actually. There are different such proteins. I just gave you one or two examples. Similarly, you have the pathogenesis related protein or the PRP protein, as I have already said, chitinase and beta-1,3 glucanase, which you can overexpress. Likewise, you have the different antimicrobial peptides, tomatin-like proteins, TLP, that actually affect the plasma membrane of the pathogen, so that the membrane becomes leaky. There is a loss of electrolyte. There is electrolyte leakage and the pathogen fails to survive. Now, another very important enzyme is the PAL, or the phenylalanine ammonia lyase. This is a particular enzyme which is the first enzyme in the phenylpropanoid pathway. And this phenylpropanoid pathway has an immense implication in the synthesis of various secondary metabolites like flavonoids, anthocyanins. So you can phenolics. So you can actually overexpress these metabolites, or you can induce the formation of these metabolites by cloning this particular enzyme. So in that way, the plants will be better able to synthesize these secondary metabolites, and it will defend itself against the pathogen. So you can go for overexpression or cloning of this PAL gene as well. Similarly, for the viral resistance, actually you can go for an approach which is called RNA silencing. I will talk on RNA silencing a little later. That is antisense RNA technology, and using this antisense RNA technology, you can generate viral resistant plants. For instance, you can develop an antisense RNA against the coat protein, viral coat protein, and you can clone that particular antisense gene, and you get a viral resistant plant. Similarly, people have shown that there is an antisense gene SAH1, which actually provide resistance against a wide range of viruses. Similarly, AC2 gene of tomato golden mosaic virus. So tobacco plants, if you transform with this AC2 gene, so you find that if you clone this gene in an antisense orientation, then you find the viral resistant plant. So all these suggest that the transgenic plants can be generated which are tolerant or which are resistant to the pathogens, different pathogens, including the insects. Now, next point is the biopharmaceuticals. Now, this is very interesting. Certain pharmaceutically active compounds can be generated in plant. All of us know that in response to antigen, our body produces antibody to defend against or to resist that antigenic attack. Now, these antibodies can be produced in plants. Plants can be exploited for the large-scale production of antibodies because they can assemble the heavy and light chain. We know that the immunoglobulin molecule, it has the heavy chain and light chain. So if you clone the heavy chain into one plant system and the light chain into another plant system and then try to cross them, then in the F1 hybrid, you will generate transgenic plants which can assemble the heavy and light chain, and you get a functional antibody in the F1 hybrid. So the advantage is that you see the plants are even capable of producing medicines for us. So you can generate a transgenic plant which is producing pharmaceutically active chemicals for you. And also the extraction of this antibody is very much easier. The stable stocks of antibody producing plants can be maintained for generations. Okay, so some of the examples are also given here, like CaroRx. This is the advanced product, and it is a dental caries preventing immunoglobulin molecule which has been produced in tobacco, and this has already undergone clinical trials in humans, and people have found that it is giving desirable results. But mind that this CaroRx product, it has been generated in plants, but it is an antibody. So you are getting functional antibodies in plant system. Of course, there are certain limitations here because the glycosylation pattern in the plant and animal systems, they are similar in the early steps, but in the later steps they are different. So you have to take care of that. So you have to actually engineer the plants in such a way so that they mimic the animal glycosylation pattern. They reflect the animal glycosylation pattern. Moreover, sometimes you need to avoid or you need to eliminate this probability of glycosylation, and therefore sometimes the peptide recognition sequences for N-glycosylation, these need to be removed. Also, sometimes you remove these chances of modification by including certain sequences, like as I've shown it here, KDEL sequence. Why you are including this sequence? So that it you are retaining this particular antibody in the endoplasmic reticulum, so that there is no modification by the Golgi bodies. So you try to retain them in the endoplasmic reticulum cell organelle. So what the process by which you can do it is through the incorporation of this sequence. So the take home lesson is that the plants can be made to synthesize antibodies. And then edible vaccines. The concept of edible vaccines is that you don't need to take medicines. You simply take genetically engineered plant. I give you an example. Like people who are diabetic, they take insulin. But if we can generate this insulin in the plant system, in fruits, so whenever you are taking any fruit, okay, like banana, you are consuming a fruit, you are actually consuming the medicine as well. That is, you are taking the insulin gene. So you don't need to be injected, or you don't need the other insulin medicine, because often you have the storage problem. Insulin needs to be stored under cold condition. So those kind of probabilities or those kind of possibilities can be eliminated by taking genetically engineered fruit which is rich in this kind of pharmaceutically active compounds. So with that, I take a break for maybe 5 minutes, and after the break we will again join for the next part of the discussions.
Do you wish to be part of a television studio like this, or any other media system, or already part of any media system, want to enhance a new set of skills? Welcome to School of Journalism New Media Studies. Here in the School of Journalism New Media Studies, we are offering program for all set of people to learn and enhance the media skills. The vision of School of Journalism and New Media Studies is to generate a new set of professionals in the area of media and communication studies with understanding about role of media in the national development and global understanding. We offer innovative programs in the field of journalism and mass communication that sets the benchmark of high quality learning and teaching process at the national level. We offer these programs across the country with the help of our regional network as well as study center. Same manner, many of our programs are offered through online mode with a sophisticated learning management system. SOGNMS offers programs from PhD in journalism and mass communication till certificate level. We have masters in journalism and mass communication. The eligibility is bachelor's degree, and we are introducing this program in 3 languages: Hindi, Tamil and English. Students from national and international students can join either through online mode as well as in the distance mode. We have 4 postgraduate diploma programs, specialized areas in the field of development communication, digital media, electronic media and journalism and mass communication program. And all these 4 programs are available in distance mode. 2 programs are available in online mode, development communication and digital media. And we have specialized certificate program in community radio. IGNOU employs various media and communication tools for better teaching and learning process. School of Journalism and New Media Studies utilizes this media system for delivering all our programs. Come and join with us for exploring a new career and enhance your media skills.
Welcome you all after the break. Now, before break, if I recap, we were discussing on the abiotic stress tolerant plant, the biotic stress resistant plant, and then we talked a bit on the biopharmaceuticals. There are different other examples as well, like bioplastics. These could also be overexpressed in plant. The environmentally degradable plastics, because nowadays we are having a lot of concern about the plastic usage. So biodegradable plastics can also be overexpressed in plant system, and people have actually done that, and they are getting fruitful results. Now, the next aspect of these genetically modified crops is biofortification. What is meant by biofortification? That means what is meant by fortify? Fortify means enhancement. So, what you are enhancing? You are enhancing the nutritional value of a particular crop. So, I will take two cases here. One is golden rice and another is the ferritin rice or the iron enriched rice. Now, what is the problem with rice? Normally, we consume rice as our staple food, but mind that we take polished rice, and polishing actually has a lot of demerits, like it removes the outer layer of the endosperm, and which we call as the aileron layer. So, normally this is being removed. Why this is removed? Because if it is retained, then there will be spoilage of grains due to oxidation of oil. And so, if you remove this layer, then that particular removal actually leads to better food storage, better grain storage, number one. Number two is it also gives a very pleasing appearance to the rice grain if you polish it. So, that is why normally the rice that we consume is the polished rice, and removal of this outer layer leads to the loss of several micronutrients. We all know different micronutrients, these are very much essential for our health. But what happens if you remove this outer layer? Along with the outer layer, these micronutrients are lost. Which are those micronutrients that are lost? Vitamins, different trace elements, especially iron. So, these are being lost, and that is why the rice that we consume, the polished rice that we consume, lacks these micronutrients. So, in that way, it has to be supplemented. These kind of micronutrients need to be supplemented in rice. The first case is the golden rice. Why is it called golden rice? As you can see in the picture, it gives a golden yellow color to the rice grain. Why golden yellow? Because you are generating rice, transgenic rice, which will be rich in carotenoids. Why carotenoids? Because carotenoid, these are actually precursors for vitamin A. What is the utility of vitamin A? You can see the vitamin A, the effect of deficiency of vitamin A. 124 million children are vitamin A deficient, and that causes about 500,000 children to go blind each year, and you have the color blindness. So, therefore, it is very much essential that you engineer rice which will overproduce vitamin A in the endosperm. In order to, so the concept is that because along with the staple food, along with rice, you are taking these micronutrients, because there are many people, poorer people, for them it is not possible to take fruits or even vegetables. Okay, so the simple staple food, they can take this staple food, and along with that they can consume this kind of micronutrients. Okay, so that is actually the main motto of this kind of work. Now, if you see rice does not contain any provitamin A in the endosperm, neither beta carotene. It does not have this kind of metabolites. It is not rich in these metabolites. So, if you need to really overproduce vitamin A, or if you want to overproduce carotenoids in rice, you have to first know what is the pathway. So, actually this beta carotene is being synthesized from GGDP, that is geranyl diphosphate, and you can see this particular pathway, and there are several steps involved in this biosynthesis. Like it starts from GGTP, then there is the phytoene production through phytoene synthesis. Then from phytoene you have this lycopene synthesis. okay, through different enzymes like phytoin desaturase, as you can see these enzymes. And then from lycopene you have the carotene, either the beta carotene or alpha carotene, depending on the presence of lycopene beta cyclase or lycopene alpha cyclase, or you can also mention it as alpha beta lycopene cyclase. So this is the entire pathway. So if you need to overproduce beta carotene in rice, you have to select all these genes, you have to clone all these genes in rice, and then only it will be capable of producing this beta carotene. So it is a quite tedious work, and this has in reality been done by a lot of research groups all over the world, and they have found that you can substitute these enzymes like phytoin desaturase, zeta carotene isomerase, zeta carotene desaturase. You can substitute these enzymes with one single enzyme which is designated here as CRT1, and this CRT1 actually stands for carotene desaturase. So people have selected this gene from the bacteria, that is Arvinia euredovora, and people have cloned these three genes, phytoin synthase, CRT1 gene and lycopene beta cyclase. These three genes have been cloned in rice, and then they could generate this golden rice, that is rice which is rich in carotenoids. So of course it's a very it's quite a delicate kind of work. It's quite a cumbersome work, and it will take time because you have to clone all these genes in vectors under specific promoter, and mind that in this case you have to use endosperm specific promoter, because you want the beta carotene to be synthesized only in the grain and not in any other tissue, and for that you have to take endosperm specific promoter. Okay, so in that way people have cloned, and the ultimate end result was that 1.6 microgram per gram endosperm. This was the amount of the beta carotene that was actually produced in the transgenic rice or in the golden rice. Of course, people went on different other modifications, and finally up to 37 microgram per gram of carotenoids they could be generated in case of rice grains. So that was the ultimate improvement in the production of the beta carotene in transgenic golden rice. Another example is high iron rice. Why iron? Because iron is also a micronutrient which has the population, human population, they have a lot of deficiency in iron, particularly the women who suffer from anemia because of poor iron levels in the foodstuffs. Like if you see, rice consumption per capita per day ranges from 300 to 600 gram in Southeast Asia, and iron requirement for adult female is 15 to 18 milligram per day. The target iron concentration in rice ranges from 7 to 14 ppm in the polished grain for individuals that consume 600 gram or 300 gram per day respectively. So of course rice does not have that much of iron which is actually needed by the human systems, and that is why people have gone for the overproduction of certain proteins which are known as the ferritin. As you can see in the figure, you just concentrate on the figure that this is known as the ferritin protein, which has iron clusters actually as mineral inside the ferritin, and people have actually overexpressed this ferritin. It is a ubiquitous iron storage protein, and people have actually overexpressed this protein, and they have got iron rich rice. The next aspect that I am going to discuss is fruit ripening. Now all of us know that all of us are familiar with tomato. So we have seen unripe tomato, which is normally green in color, okay, or yellowish or greenish yellow, and the ripe tomato which is actually red or orange in color. So whenever a fruit ripens, fruit like tomato ripens, there are certain changes associated with this ripening process. Like there is a texture change, there is a softening. That means if you have the unripe fruit, it is hard in texture. If you have the ripe fruit, it is soft in texture. Then there is a cell wall disassembly. Why it turns soft? Because there is a degradation of cell wall. Because the pectin is being degraded, as a result there is a softening. conversion of starch to sugar, you have a high amount of soluble sugar. If you taste an unripe fruit and a ripe fruit, you will find that the ripe fruit is comparatively sweeter than the unripe fruit. Alterations in the pigment biosynthesis, there is a lot of flavored compounds generated, and the red color that is generated, it is because there are certain pigments, okay, the chlorophyll is being degraded to give rise to the other pigments. That's why there is a change in color. And last but not least, there is an induction of a phytohormone called ethylene. We all know that ethylene induces fruit ripening. Now, if we go through the basic plant physiology, then we can see that ethylene is actually produced from it is produced via this pathway, as I have shown. You can see on the left side that it actually the methionine is the precursor, and from the methionine there is first the conversion to ACC through ACC synthase, and then ACC it undergoes oxidation to ethylene through ACC oxidase. And because of this induction of these enzymes or these genes, ethylene is being produced. So if you want to delay the fruit ripening process, because you want to increase the shelf life of the fruit so that the fruit does not get rotten easily, so what will you do? You will try to stop or retard the ethylene production. So you will try to reduce the expression of ACC synthase or ACC oxidase, and people have actually done that through one technology which has been shown here as the antisense RNA technology. That means instead of taking the normal gene, you take the antisense version of this gene and you try to clone the antisense RNA. You try to generate the antisense RNA or the antisense version of the gene, so that the sense RNA and the antisense RNA, they will form an RNA-RNA hybrid, and thereby they will lead to the silencing of the gene or non-functional gene. So in that way, you can make the ACC synthase or ACC oxidase non-functional, and you can see the effect in this very diagram, that when people have actually generated antisense ACC synthase, there was a severe inhibition of ethylene synthesis and the fruits remain healthier for a longer time. Control fruits, if you place them, they undergo food spoilage and they ripen within a short time, whereas the antisense fruits, they ripen at a much longer time period. That means the fruits remain healthy, they remain unspoiled for a longer time, and in that way you can increase the shelf life of the fruit. Similarly, if you see the ACC oxidase, people have also undergone antisense versions of the ACO, that is ACO that is oxidase gene, and similarly they have seen that the antisense version of this gene, it leads to 97% inhibition in ethylene production. Another gene which people have cloned, perhaps we have heard about the flavored sour tomato. What is flavored sour tomato? It is a genetically engineered tomato where the polygalacturonase gene has been downregulated. What is the function of this polygalacturonase gene? Polygalacturonase gene is a gene which actually degrades the polygalacturonic acid, which actually makes up the pectin. So the pectin degrades and therefore the cell wall gets softened, and that is why the fruit turns soft at a much earlier rate. So what you can do, you can just silence this polygalacturonase gene through antisense RNA technology, and the transgenic tomato fruit that you derive is known as the flavored sour tomato. So it has been found that the fruits, they remain healthier. There is lesser spoilage of the fruit. Mechanical handling becomes easier. The fruits do not get destroyed or ruptured easily. So that kind of work has been done and the flavored sour tomato has been generated. So this is nothing but this is a genetically engineered tomato which has been basically generated for increasing the shelf life of the tomato fruits. The next application is genetically modified C4 rice. Now C3 and C4 photosynthesis, this is one of the very important modules that perhaps you have studied in the under plant physiology. Now why C4 plants are more advantageous? Because they have reduced photorespiration and they are photosynthetically more active. The question is that C3, like rice is a C3 plant. So obviously it will have lesser photosynthetic efficiency, even wheat. So they will have a lesser photosynthetic efficiency than the C4 plant. So whether it is possible for us to genetically engineer the C4 genes or the C4 enzymes in rice or wheat so that they can be made to behave as a C4 plant. The predicted atmospheric carbon dioxide concentration will be 520 to 550 ppm in 2050. So it is rising, and it will continue to rise. So achieving a 40 to 50% increase in yield is probably beyond the capacity of rice. However best we want, we will not be able to go for the increase the production of rice with its current photosynthetic pathway or photosynthetic efficiency. So we need to increase the photosynthetic efficiency. Achieving yield beyond 12.5 tons per hectare in the next 50 years would require the introduction of C4 photosynthetic pathway in rice. Now, the learners, one question might come in your mind that if you have studied C4 photosynthesis very carefully, you might have seen that they are the pattern of, the spatial pattern of distribution of enzymes was different, because there was the mesophyll cell and the bundle sheath cells, and there were some specialized enzymes like phosphoenol pyruvate carboxylase, pyruvate orthophosphate dikinase, and the NADP malic enzymes. So this is actually the way by which the C4 photosynthesis operates. That is, some reactions occur in mesophyll cells and some in the bundle sheath cells. So a question might come in your mind that this is not there in the C3 plants. Then how the C3 plant will act as a C4 plant? Now in this context, I should mention that there are certain plants which do not have the Kranz anatomy, which do not have such spatial distribution of enzymes, but even then the C4 systems lacking Kranz anatomy is also there. Okay, there are several C3, C4 intermediate species. Those are available. So this gives us an impression that probably the C3 plants can be made to behave as a C4 plant. And people have actually done that. People have actually generated the C4 plants, C4 rice, by cloning or by introducing the C4 enzymes or the C4 genes in rice. And this kind of research is still going on, and they are trying to develop the C4 rice. So this is another area in which in the coming years, maybe we will have more improvement in this particular area, okay, in this particular area of genetic engineering research. Next, we have the herbicide tolerance. Now, what is meant by herbicide? Now, in the agricultural field, we are interested only with the agriculturally important crop. We don't want the weeds, okay, unwanted weeds. So what do? We apply herbicides or the insecticides, herbicides. But what is the problem? Problem is these herbicides, they also affect the agriculturally important crop. That means not only the weeds, but also the agriculturally important plants will also be affected. So what we have to do? We have to actually make the agriculturally important crops herbicide tolerant, so that they are not affected by herbicides. Only the weeds or the non-desirable plants, they will be affected. So for that, what we have to do? We have to again select few genes, like which are the herbicides that we normally apply: bialofos, phosphinothrycin, glufosinate, ammonium. These are some of the herbicides. They inhibit the glutamine synthase enzyme, and therefore they result in the ammonia accumulation, okay, and therefore the plants die. So what do? In this case, we have to clone the different genes, like BAR. It is coded by phosphinothrycin acetyl transferase enzyme, which converts bialofos to a non-herbicidal acetylated form by transferring the acetyl group from acetyl-CoA on the free amino group of phosphinothrycin. So what we have to do? We have to clone this enzyme so that the herbicide is detoxified, and so the herbicide will not affect the agriculturally important crop. So that is actually the main intention, or that is actually the main purpose of this kind of work. Okay, so you can try this with the other herbicides as well. So you can clone any herbicide tolerant gene in the agriculturally important crop, and so those crops will not be affected by herbicide. You don't clone those genes in the weeds, so that the weeds will be affected by the herbicides. So that is actually the main purpose of doing this. Now in floriculture also, like if you want to develop new colors of flowers, then you can use this plant biotechnology or genetic engineering, like variegated flowers. Chalcon synthase gene or the enzyme has been overproduced if you want to get white and variegated flowers, like in petunia, chrysanthemum. So in floriculture, okay, and in the creation of different ornamental plants, you can use this plant biotechnology. We are almost towards the end of the lecture, and the last of the application that we are going to discuss is the male sterile transgenic plant. Now what is the utility of male sterile plants? Now whenever one of the serious issues of genetically modified crops, why people are raising protest also, is that because of the pollination, because of the transfer of the pollen grains from the transgenic crops to a wild type crop, there will be a contamination. That means which are non-transgenic plants, they might get contaminated because of the pollen transfer or because of the pollen escape. Now pollen transfer, that means mostly pollen, you all know that it is generated within the anther, and that is the male reproductive hole. And so if you can generate a male sterile plant, then there is no question of this pollen escape or pollen transfer. So that is the main fundamental or what should I say, purpose of doing this work, to generate a male sterile plant. Now that means you have to do something so that you generate pollen grains which are sterile, which are non-functional. So cytoplasmic male sterility is associated with defective functioning of the tapetum of the anthers that supply nutrients to the pollen grains. So tapetum layer in the anther, what is its function? It provides nourishment to the pollen grains. Now you choose one gene which is called Barnes, and that the source of this gene is a bacteria, Bacillus amyloleucofaciens. So you clone this Barnes gene under a tapetum specific promoter, okay. So it will kill only the tapetal cells, preventing the pollen development. So you generate the male sterile lines, okay. But you might require to restore the male sterility as well. That means you might want to restore the fertility in the plant. For that, what should you do? You choose another gene which is called Barster, and you overexpress this Barster gene again with the same promoter, that is the tapetum specific promoter, and in that way you can restore the fertility also. Okay, so the concept of male sterility, that is male sterile plants can be generated using the Barnes gene, and if you want to restore the fertility, then you can use the Barster gene, and if you cross these two, then you can actually generate this fertile plant. So we are almost towards the end, and I summarize now. Okay, I summarize this particular entire discussion, that is biotechnology, as it has an immense implication. As you have seen, in several areas you find the implication of genetically engineered crops, and in fact the first event of the transgenic that was generated was with regard to herbicide resistance. Though I talked on herbicide tolerance or herbicide resistance almost towards the end, but you have to understand this was probably the first achievement of the plant biotechnology. And then people detoxified the herbicide, especially in the agriculturally important crop, and people have derived desirable results. And then resistance to the insects, particularly I have talked today on the BT transgenics, in order to protect the plants against the Lepidopteran insect larva. This I have talked. Then the generation of the abiotic stress tolerant plant, environmental stress tolerant plant. Okay, different types of environmental stresses are there, like salinity, drought, cold, heavy metals. So you can generate resistant plant. Okay, and then you can generate plants which are desirable in agri-horticulture, floriculture. Okay, you generate flower saver tomatoes which have delayed ripening. Okay, so you can increase the shelf life of the fruits. You can generate the C4 rice, that means the C4 genes or the enzymes can be overexpressed in the C3 plants as well. So you see that you have a lot of implications, okay, in the improvement of the crop. Now of course there are different biosafety issues, ethical issues, associated with these transgenic crops, which I am not going to talk today. Maybe in some other discussion I will take up this topic. It is also a very serious topic which needs discussion and our attention. What are the biosafety issues and how these biosafety issues of the transgenic crops are being addressed, or how these are solved. So those probably I will take up in some other discussions. So I hope I could make this particular unit somewhat clear and lucid to you. If you face any question, please feel free to ask any question, or any of your queries are always welcome. So thank you all for listening to me, and have a very, very happy learning.
Are you a fresh graduate? Do you want to fulfill your educational aspiration by pursuing higher education in various interdisciplinary